Welcome to Unit 6, Gene Expression and Regulation. In this video, we're going to be looking at Chapter 13, the molecular basis of inheritance. How are traits passed down from parent to offspring? But what is the molecular basis? What is the molecule behind it? In this, you must know, or in this video, you're going to be learning about, you must know, number one, the structure of DNA. This will be throughout the video, but really focused near the end of the video. And then in this video, we'll look at the history of DNA, how the knowledge about how DNA was gained from the work of Griffith, Avery, McLeod, McCarthy, Hershey, Chase, Wilkins, Franklin, Watson, and Crick. So all these names, all this research that was done to discover what does DNA actually look like, because we cannot see it with our eyes, we cannot see it with a microscope. So how do we know what the structure looks like? And when did we discover DNA? Those questions will be answered in this video. Chapter 13, we'll talk about DNA, the genetic material. This beautiful molecule here, deoxyribonucleic acid, which is essential for life. If you're alive, you have DNA inside of you. And every living thing has a molecule very, very, very similar to this one right here. And this molecule is the instruction manual for life. And this molecule is the reason why life can continue on planet Earth, because this molecule can be passed down from generation to generation. So how is it that the DNA inside of you is connected to your ancestors and people that lived hundreds of thousands of years ago? What is the connection between this physical molecule? It's something that you can actually touch. It's made up of atoms, and this molecule is somehow connected to the past. This is such a beautiful concept to me, and I hope that you enjoy learning more about DNA in this unit. But for us to understand DNA, we need to understand the history of DNA. We start in the 1850s with Gregor Mendel, who kind of had this idea that traits are passed down from parent to offspring, and he did his famous pea plant experiments and found that some of these traits are passed down, and he had this idea that maybe these traits are connected to parents because of some sort of allele or some sort of factor that is passed from parent to offspring. We're going to look at Morgan, Griffith, Avery, McCarty, McLeod, Hershey, Chase, Watson, and Crick. So these names are going to come up throughout this lecture. And it's just important for you to kind of see the story of how DNA was discovered, because this is such a recent discovery. And in the 1950s, you really kind of put all the pieces together. And this is so important because this discovery is so new. There's so much more there is to uncover and unpack in the scientific community. So I want to encourage you to think about the implications of this. How is this relevant to you today, our understanding of DNA? And when do you think about DNA? When do you think about your DNA? Why would you need to know about your DNA? So have these things in mind as you continue watching this video. I want to just start with this quote. I encourage you to pause the video and just read it. Take a moment to read this, and it'll kind of set the tone for our little history lesson on DNA, and how we really use the knowledge that has been discovered before us to be able to see the world more clearly and to basically understand in more detail what was known in the past. All right, think of science as kind of like uncovering like a painting. Like you're able, maybe in the past we were like really zoomed in on one part of the painting, but as we've been able to uncover more and create new discoveries, we're kind of stepping back and back and back, and we're able to kind of see the picture fully, whereas in the past we were only able to see one piece. All right, so the first piece of history, we're going to talk about Morgan, and we actually talked about Morgan last chapter. He worked with Drosophila, or fruit flies, in the 1930s, and he found that genes are on chromosomes. So that was his big discovery. But we know that chromosomes are made up of two molecules. They're made up of proteins. So here in pink, we have these proteins called histones, and DNA, deoxyribonucleic acid, and nucleic acid, which is a long string molecule that's wrapped around these proteins. Now, scientists weren't sure if the information, genetic material, was found in DNA or in proteins. They knew what the molecules looked like, but they weren't sure what was actually the piece, the information, the code that was passed down from parent to offsprings. And even through the 1940s, scientists thought that proteins were genetic material. Now, this doesn't make sense to us because we know DNA is our genes. We connect those words. But in the past, they knew that there was this physical chromosome that passed down traits from parent to offspring. You can see chromosomes under a microscope, but they didn't know where the information was actually found, and they didn't really know what it would look like if you were to zoom closely into this chromosome. So what we're going to be exploring in the next few for the next few minutes is, okay, did scientists discover that proteins contain genetic material or DNA? And so we'll think about maybe first, why did scientists think in the 1940s that proteins were the genetic material? Well, the reason they thought this is because there are thousands of different types of proteins with different shapes and structures and functions. And that seems like a good molecule to pass down genetic material, right? Think about how every living thing is so different, so diverse. And so maybe these different types of proteins can pass down different instructions for different traits for living things. So that's kind of one of the reasons why scientists thought proteins were genetic material, because you can build up so many different structures, so many different types, and that was kind of the idea before DNA was more well known. Okay, so we're asking this question: is it protein or is it nucleic acid, the DNA piece? So let's take a look at this scientist, Frederick Griffith. He was actually working with bacteria, and his job was actually to find a cure for pneumonia. So he worked with a strain of bacteria called Streptococcus pneumoniae, and this bacteria, he was trying to figure out how it's connected to pneumonia. And he did this experiment where he actually took this bacteria and he mixed it with other bacteria and he injected it into living mice. And he found that if he took a piece of bacteria that was harmless, that basically wasn't going to infect the mice with a disease, it was harmless. It was still bacteria, it was still a living organism, a prokaryotic organism, but it wasn't harmful. So this harmful live bacteria, it was mixed with some infectious bacteria. So there are some bacteria cells that actually can cause infections. But what he did was he killed it with some heat. And he found that when you mix these two, it causes a disease in the mice. So we'll take a look really closely at this experiment. And he found that there was a substance passed from dead bacteria to live bacteria. So for some reason, bacteria, even if it's alive or dead, they can pass something between each other. And he called this a transforming factor. So transformation is this idea that there is something in a cell, so for example, a bacteria cell, that can be passed or transformed to another bacteria cell. And it doesn't matter if that bacteria cell is alive or dead, there is something inside of the bacteria that can be shared or swapped or passed or transformed from one to another. So let's take a look closely at his experiment. So here we have one test, and we took a pathogenic strain of bacteria. Pathogenic means it's going to cause illness to the mice. So this is a form of harmful bacteria. So a strain is just like a type of bacteria or species of bacteria, and this was harmful. And these are found in bacteria cells, right? Inject it into the mice, and all the mice die, which makes sense. This is pathogenic. It's going to cause harm. Then he took a strain of non-pathogenic bacteria. So this is like safe bacteria, healthy bacteria. It's not going to hurt the mice. And of course, the mice live. Then he took the same pathogenic bacteria, but then he heated it up really, really high, denatured enzymes, and killed off the bacteria, injected it into the mice, and the mice live because the pathogenic bacteria is dead. It's not going to be harmful anymore. But finally, he took the heat-killed pathogenic bacteria. So these are, right, you've killed off the bad stuff, and the non-pathogenic bacteria. Injected it in, and the mice die. So what did he find? He found that there's something about this pathogenic bacteria that can be passed to the non-pathogenic bacteria that cause the mice to kind of pick up something that the pathogenic bacteria had before. There's something in this heat killed bacteria that could still be passed on to the living non pathogenic, the healthy bacteria, and cause the same disease killing properties. So he calls this transformation, the ability for something to be passed between one bacteria cell to another bacteria cell whether or not they're alive or dead. So the people who worked on this experiment, Oswald Avery, McLean McCarthy, and Colin McLeod, these are the people who came up with this idea of the transforming factor. Something can be sent between one bacteria to another. All right. So like we said, kind of to go over this again, these scientists, they purified DNA. So they pulled out DNA just on its own, and they took out RNA, ribonucleic acid, which is another type of genetic material. But at the time, they didn't know that. They just knew that this was a molecule, and proteins. And they took this from pathogenic, from harmful Streptococcus pneumoniae. This is a species of bacteria, this strain. And then they added them to different like batches. So, for example, they took the Streptococcus pneumoniae and they added everything, DNA, RNA, except for protein. So they added DNA, RNA to this bacteria, and they didn't add protein, but the bacteria still transformed. So even without protein, there was some sort of transformation happening, something being passed between the bacteria. Then they took bacteria and they added DNA and proteins, but no RNA. And they found that the bacteria was still transformed, even without the RNA. And then finally, they took RNA and proteins and they added them to some bacteria, but they found that the bacteria did not transform. So nothing was passed between. And so what this kind of led the scientists to conclude is that, okay, what is the thing that is actually being passed between bacteria? Well, it might be the DNA, this deoxyribonucleic acid, because without the DNA, they found that nothing was passed between cells, between bacteria cells. All right. So again, one more experiment done by Hershey and Chase. We call this a blender experiment. And what happened here is we take this virus here called a bacteriophage. Phage means to kill, and bacteria. So these are bacteria killers. These are viruses, and they have this kind of crazy shape here. Now, if you look closely here, in the center of the head of the bacteriophage, this virus here, there is DNA. And then you have kind of this collar, this sheath, and then these tails. It looks so interesting. It looks kind of creepy here, but this is a virus, one virus. And the outside here, the head, the collar, the sheath, the tail fiber, this is all made up of protein. But inside, it's made up of DNA. And so here's what the scientists did. They took phage viruses. So this is a bacteriophage. You can shorten it as phage. They took this virus and they put them in two media. Media is kind of like a culture or an environment where the phages can grow. Now they took one group of phages and they added this thing called an isotope or a radioactive label. Now here's what that means. You can actually take these atoms here, sulfur or phosphorus, and you can add them in a mixture with bacteriophages like you see here. And you can see, for example, if you take a batch of bacteriophages and just add this sulfur isotope right here, sulfur 35, if you add the sulfur to the bacteriophage, some of that sulfur will be mixed together with the bacteriophage and it will be attached to some part of the bacteriophage. So imagine if you took sulfur and mixed it with this, the sulfur will actually become attached to some part of it. It'll become incorporated with this bacteriophage. Then you can take another group of bacteriophages, or phages for short, and you can mix them with some phosphorus isotope, this radioactive isotope. And then that phosphorus will become incorporated. It will actually stick onto this bacteriophage in some way. Now, it's important to know that these bacteriophages, once they're labeled with these isotopes, they will actually glow. You'll be able to see this glow here, and you'll be able to kind of see or be able to measure where did that isotope attach onto the bacteriophage, okay? Now, why did we use in one group sulfur? So one group of phages with sulfur and one group of phages with phosphorus. Well, here's the reason. Sulfur is an atom that is found in proteins. Okay, so if you were to look at a protein structure, or back to unit 1, an amino acid, you would see the central carbon with a hydroxyl group, and then an amino group, and then a hydrogen on one side, and then attached to that there's an R group, and that R group often has sulfur. in that R group. So that sulfur will be able to be attached to proteins. Phosphorus is an atom found in DNA. And we'll learn more about this later. But phosphorus is an important part of the DNA molecule. And so phosphorus can attach to DNA. So what the scientists were trying to figure out was if you mix this phage with sulfur, where do you think the sulfur will attach? Will it attach to the DNA or will it attach to the protein exterior? In the same way, the phosphorus, will it attach to the DNA or will it become incorporated into the protein exterior? So let's take a look. Oh, and the cool thing about bacteriophages, these viruses, we can kind of see a picture here, is what they do is they actually infect bacteria, right? They kill bacteria. And here's how they do it. So here's the structure, and they actually don't enter the bacteria at all. Here's a bacteria cell down here in the bottom. But what they do is they insert their DNA into a bacteria cell, and that DNA is actually going to be used by the bacteria cell on accident to build proteins that are going to destroy the bacteria cell. And so that's how the virus destroys this bacteria. But as you can see, the protein stays on the outside and only the DNA, right? Only this molecule will go in, not the protein. So the bacteriophage exterior, this like skeleton exterior, cannot enter the cell. Only the DNA can get in. Okay, so let's take a look a little bit closer at this experiment. I know this is a lot of detail, but I want to encourage you to focus in here because you're going to be able to understand it better after this. Okay, so two experiments, two groups. So first we take our phages and we add them with the sulfur isotope, okay? Then we take the phages and we label them with phosphorus isotope. Okay, so two different labels, two different groups here. Now, what they did was they took them and they added them to some bacteria cells. You can see the bacteria cell here. And then they agitated the bacteria and the phages. Agitation means they put it in a blender basically, or something called a centrifuge, and they shook it up so that the phages would actually be released and fall off of the bacteria. But before the phages are released, they actually are going to insert their DNA inside of the bacteria. Insert the DNA inside here. Now, at the time, the scientists didn't know that this was happening. They had no clue what was going on. But what they're going to be looking for at the end of the experiment is to see, okay, where did our radioactive isotopes end up? When they see this glow, they can basically follow the sulfur and the phosphorus. Where does it end up? Where does it end up attaching to? So when they put the bacteria cells and the phages into the centrifuge, they shook it up. The exterior, the phages fell off, and what was left inside was the DNA. Same thing over here. The phages fell off, and what was left inside was the DNA. Now, in the group of bacteria cells and phages, that was labeled with sulfur. Remember, sulfur can only attach to proteins. The sulfur was only incorporated or labeled into the protein exterior. It wasn't able to attach to the DNA because the sulfur doesn't become incorporated with DNA. And so what happens is the bacteria cells, once they got removed from the exterior protein, they did not glow. Right? There's no ability to see anything in the cells because they're not labeled with anything. Only the exterior pieces were labeled. However, in this one, because you mixed it with phosphorus, the phosphorus was incorporated with the DNA. It can stick to the DNA. The DNA enters into the bacteria cell here. The protein exterior, that's kind of the shell or the skeleton of the bacteriophage, will be removed after the centrifuge, after the blender. And so then what is being able to be seen, what is radioactive now, what is glowing here is the DNA. And so the bacteria cells will glow because they have been labeled with phosphorus, which is attached to the DNA. So this led the scientists to see that what is it that's actually the transforming factor? What is it that can be passed from one to another? It's not the protein exterior, but it's actually the DNA that can be inserted. And again, they found this by labeling the DNA. Labeling means they added this special marker or this molecule that can glow, and then they were able to track it and trace it as it goes into the bacteria cell and not left over in the outside. Okay, so once again, what is found inside the cell is our DNA. So that's the thing that can be transformed. That's the thing that can be added into the cell. Okay, now in this group, we found sulfur radioactivity, so that glowing, there's radioactivity found in the liquid medium, so the outside of the bacteria. But in this one, radioactivity was found in the bacteria cells inside. So which molecule carries the viral genetic info? Which molecule is actually able to be passed from one to another? That is the DNA. Okay, so here are our famous experimenters, Martha Chase and Alfred Hershey, who did these experiments. And just another close-up look at what's going on here. Let's kind of follow the two experiments once again with kind of some different details here. So step one, here's our medium, and we have in here radioactive sulfur. So the sulfur isotope in pink. And we mix inside some phages. So the phages, the bacteriophages are mixed with sulfur. And the phages are going to infect the bacteria cell. So here in orange is our bacterium, our bacteria cell, and then the phages will insert their DNA only, nothing else. Now, a protein, because it's been labeled with sulfur, is the radioactive part or the glowing part. Now, step two, you're going to agitate to separate the phages on the outside from the bacteria on the inside. So they basically fall off. And what happens is you form a pellet at the bottom of the test tube. This pellet is going to just contain the bacteria cells because now you've separated the bacteria cells from the rest of the medium, and this is just full of liquid with empty protein shells. Now, what is radioactive or what is glowing here? The scientists are going to measure the amount of radioactivity, and they find that only this liquid here on the top has radioactivity and not the pellet, which shows that what was labeled was the protein and not the thing that was entering into the bacteria cell. So the stuff on the outside, the protein on the outside, was able to be incorporated with the sulfur. Okay, and then this is second experiment. We're going to mix our phages with phosphorus, radioactive phosphorus. And that radioactive phosphorus will be incorporated into just the DNA piece. Now, when the phages infect the bacteria cells, they're only going to put their DNA inside, not the protein exterior. We're going to centrifuge, we're going to separate, we're going to blend out the proteins so they fall off and just have the radioactive DNA inside. And then you're going to see down at the bottom, here is our bacteria cells, which are heavier. They're going to sink to the bottom. And then the lighter liquid at the top will be left over. Now, what is radioactive here? Is it the protein or is it the bacteria with the DNA inside? It is the pellet, or it is the cells are now radioactive or glowing. And so the scientists have traced, they've seen, oh, the phosphorus is incorporated into the DNA, and that is the thing that is actually going into the cell. That is what's being transformed into the cell. Okay, so these are the famous Hershey and Chase blender experiments. And here's kind of like a summary. Like I said before, you can pause this and review it if you need to. All right. So what does DNA look like? In this next section of the video, we're going to be looking at lots of models and artistic depictions of DNA. And I'll give you a bunch of those and we'll kind of walk through the molecule. But it's really important that you're understanding this model and drawing out these models. There are a lot of different types of pictures because in some pictures, we're going to be zooming in really, really close at like the actual atoms that make up the molecule of DNA. Some we're going to be zooming out and we're going to be looking at the molecule from a little bit farther distance. And so this perspective is going to be important for you to keep track of. But DNA ultimately is a polymer. And a polymer is a molecule that's made up of individual subunits, kind of like beads on a necklace. So you have like a string of things on a necklace. And polymers are made up of monomers. A monomer is one piece or one bead of that necklace. Now, DNA is made up of these pieces here called nucleotides. So one nucleotide is one monomer, one piece. And if you put a bunch of nucleotides on a chain, that creates a long molecule that we call a nucleic acid. There are different types of nucleic acids, but DNA specifically is called deoxyribonucleic acid. Why is it called deoxyribonucleic acid? Well, that's because you see here that in the nucleotide, in this monomer here, you actually have a sugar, and that sugar is called deoxyribose. Deoxyribose because if you notice here, there is the D means not or no. Oxy means oxygen. And if you see here on this this part of this sugar, right? There should be an oxygen here, but it's missing. So it's called deoxy. If you were to have an oxygen right here, you would call it ribose or oxyribose. There would be an oxygen there. So when we're naming chemicals, when we're naming molecules, we're really talking about what are the atoms that make up that molecule. Now, a nucleotide is made up of three parts, okay? So the first part is this phosphate group, and here's that phosphorus. Remember how we talked about in the previous slides how phosphorus can actually be incorporated into DNA? Well, that's because DNA has the phosphorus atom in it, which is bonded to some oxygens. That's a phosphate group here. So this is a functional group that is made up of phosphorus and oxygen. Now, that is bonded through a covalent bond to our deoxyribose sugar. So this is a 5-carbon sugar. So 1, 2, 3, 4, 5 carbons. Now, they haven't labeled the carbons with a C at these vertices here because scientists get lazy and they don't always draw the carbons. But this sugar also has some hydrogens and some oxygens here. And then this sugar is covalently bonded to a nitrogenous base. And we call it nitrogenous base because it's made up of lots of nitrogens here. The parts that are missing, these are carbons between those nitrogens. And we've got some hydrogens going on. Now this base, notice that there are actually different bases that you could plug in and kind of pair up with this sugar here. So if it has this shape, we call it adenine. This shape, we call it cytosine. If it has this shape, we call it guanine, and this shape, we have thymine. So these look kind of similar in structure. They're all made up of some nitrogen, but they have different amounts of oxygen, different amounts of hydrogen, and they have kind of different ring structures. So one of these is called one ring. If you have two of them together, we call these two rings. Even though they're not circular, we still call them rings. Okay, so these are our bases, our sugar, and our phosphate. Now on the outside, so if this is one long chain here, notice that on the outside we have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So we call that our sugar phosphate backbone. Okay, so a backbone is like the structure that holds everything up. This is our sugar phosphate backbone. And then we have our nitrogenous bases on the inside. And we'll look more closely at this molecule as we keep going, but just kind of glance here and take a look at what this molecule looks like. Okay, so kind of continue our history lesson of DNA. Right like in the late 1940s, scientists knew kind of basically what these nitrogenous bases looked like. They had the idea of what the sugar phosphate backbone looked like. They kind of had these puzzle pieces where they knew what a phosphate group looked like, they knew what a sugar looked like, they knew what a thymine looked like, and they also knew that DNA had these shapes inside. And I'll explain a little bit later how they knew that. But they didn't know how they were pieced together. They didn't know which one went over here and which one went over here and how they were all arranged like a puzzle or kind of like Legos that you put together. So we are going to continue to look at some data. And one really important scientist that came up with some important evidence and found some data about the structure of DNA is called Chargaff. And Chargaff, we call his studies Chargaff's rules, or what he found, his conclusion, Chargaff's rules. And he found that the composition of DNA, composition means what it's made up of, varies from species to species. So humans and sea urchins, we have a little bit different composition of our DNA. It's a little bit different. And he found that all the 4 bases, right, so these bases right here, they are not in equal quantity. So all living things have these molecules inside, so humans and sea urchins, but he found this interesting thing that sea urchins and humans have different amounts of thymine or guanine or cytosine or adenine. So even though we have the same molecule, we have different amounts of each of these. So that was kind of an interesting thing he found. And he found that in humans, there was a ratio of bases that are consistent for every human that he studied. He studied the DNA of many humans, and he found that adenine always comes out to 30% of the DNA molecule. So if you were to look at the whole molecule and count up how many adenines compared to all the other pieces, 30% of that molecule is made up of A bases. Around 30% is made up of T's, 19 or 20% is made up of C's, and 19% is made up of, G's, and 20% is made up of C's. So this was kind of an interesting discovery. And do you notice any patterns in what he found in the composition of DNA? So again, if you took all those bases and looked at what percentage is found in the DNA, you'd find equal amounts of A's and T's and equal amounts of G's and C's. And if you add all of these up, you would find equals 100. Okay, so what does this mean for the molecule? We'll find out in a little bit when we kind of see how the DNA molecule is fully structured. But some more interesting data that Chargaff found is that the percentages of A's, T's, C's, and G's varies between different species. So if we have corn here, corn has 26.8% A's, 22% G's, 23% C's, and 27% T's. Okay, so notice that the A's and the T's are kind of similar in percentage, the G's and C's are similar. But notice that the percentage of G's and C's together, 46%, A's and T's is about 54. But then look at an octopus. So 33% A's and like 32% T's, 70% G's, 17% C's. But the ratio of how many G's and C's together is 35 versus 64. So you can keep looking here. Here's a human. You can see 30 and 30, 20 and 20. Grasshopper, 29 and 29, 20 and 20. So this interesting pattern that we're finding, all species have kind of different amounts of G's and C's, T's and A's, but they all have the same ratios of A's to T's versus C's to G's, right? Equal ratios of A and T, equal ratios of G and C. All right, so we'll keep that for a moment. It wasn't until actually the 1950s that all of this data was really put together and the full picture was summarized, and the final molecule of DNA was known. Okay, now these two scientists, Watson and Crick, they finally pieced together what this structure looked like, again, using the data from their predecessors. So here's like a kind of funny little graphic if you want to pause and take a look at it. Quick commercial to show you my cat, Sammy, who has been meowing at my door and wants to say hello. Okay, back to the video. So Watson and Crick are famous for developing the double helix model of DNA, which is, I'll kind of show you a picture. This right here, one of these is a helix, one of these kind of like staircases, spiral staircases. And then this right here is a helix. So there's a double helix. There's two of them that are kind of wound around each other. So Francis and Crick were the ones who put this model together. But there were other scientists working on this question, like what is the shape of the DNA molecule at the same time? Rosalind Franklin, Maurice Wilkins, and Linus Pauling, who are all kind of at the same time trying to work out what does this molecule look like. So let's first look at the research done by Rosalind Franklin. She was a scientist who studied in London, and her work, what she did was called X-ray crystallography or X-ray diffraction. And basically her job was to take a molecule and use a tool called X-ray crystallography to basically take a picture or an image of a molecule that's too small to see with a microscope. And her photo here of DNA, this is her picture she took of DNA, is one of the most famous images in all of science. This is called Photo 51. And her picture of DNA, it doesn't really look like it to you because it's harder for us to read these patterns here, these pictures of molecules called X-ray diffraction or X-ray crystallography. But this picture was able to be, basically you can take some math equations and plug them into this pattern that you see here and analyze this picture and kind of trace back what does the shape of the molecule look like, even if it's too small to be seen with the microscope or the human eye. Now let's see if I have my picture here. Let me get it for you all. So X-ray diffraction looks like this. This is what Rosalind Franklin's job was. Her job was to take a beam of X-rays and shoot it at a molecule. Again, this is too small to be seen. What happens is there's a beam of particles here that are shot at the molecule, and some go straight directly through, but others hit the molecule and kind of bounce off. We call that diffraction. they hit and bounce off. And what happens is you have this film or this kind of piece of paper on the other side of the molecule, and it's going to be basically tracing where these particles end up after they're diffracted. Here's another picture, so kind of from another direction. Here's an X-ray tube. It's shooting out X-ray particles or small particles. Here is a lead screen that's kind of filtering them in one direction. Here's a molecule or a solid that is made up of molecules, maybe like kind of like DNA. And as the particles in this X-ray pass through the crystal, they're going to be diffracted. They're going to scatter as they pass through, and they're going to form some sort of pattern, these spots on the film that the particles are hitting on the other side. And you're going to get something that looks kind of like this. And again, we can't read that pattern, but you can use really complex equations to trace back and kind of work backwards. Okay, what was the shape of this molecule? So Rosalind Franklin did this, and she came up with this picture of DNA. And it was really, really helpful for Francis and Crick in discovering the actual double helix nature of DNA. Now, the sad part is that Rosalind Franklin died when she was 38 from cancer. So she was actually, unfortunately, not able to be recognized for her research when the other scientists, Francis and Crick, won a Nobel Prize for discovering this double helix structure of DNA. All right, so looking a little bit closer at DNA, we're going to pause our history lesson for now, and we're going to now look at some examples and models of what DNA looks like. And again, there's so many different illustrations. They're all representing the same basic structure, but we're kind of zooming in or out. So here we see a space filling model. So this is like the actual 3D shape of the double helix. So we can see all the different chemicals or molecules that make up this double helix. So in the phosphate groups here are yellow. So we see kind of in this ribbon on the outside, these yellow phosphate groups, and these deoxyribose sugars are in blue. So again, remember when we talked back about the structure of DNA, we saw there is phosphate and sugar. And so that's what we're seeing in the same colors here in this model, phosphate and sugar. And those are making up this backbone of the spiral of each helix. And then the nitrogen spaces, they are in different shades of orange and green, just like we saw in the picture before. So here we have actually, we're able to measure the distance between two bases. And we know that the distance is 0.34 nanometers apart. And then for every turn, for every full turn of the helix, there's 10 base pairs or 3.4 nanometers. And so scientists were actually able to figure out these dimensions by studying Rosalind Franklin's X-ray diffraction pattern and work backwards and kind of figure out these measurements. You can kind of take a closer look here, but we call DNA the double helix right-handed because if you were to wrap your right hand around it, it would wrap nicely around this molecule, but it wouldn't work with your left hand. Okay, here is a really, really amazing image. This is going to be so helpful for you to understand. So make sure you're paying close attention here and drawing this out as you listen. So in this model, what we've done was we've actually taken this DNA double helix and we've untwisted it so that these sides here, this sugar phosphate backbone, is kind of like the sides of a ladder. And then these bases here, these base pairs, are kind of like the rungs or the steps of a ladder. So we have untwisted it, and now we have two strands. So we have one strand on the left-hand side and one strand on the right-hand side. And we're going to be looking at the chemical details. Now notice that the two strands, and here's a key vocab word, they are anti-parallel. We're talking about the structure of DNA. Anti means opposite or against, and parallel means they're next to each other. They're going in the same direction. But notice here that we have the phosphate and the sugar kind of facing in this direction, and then the phosphate and sugar facing in the opposite direction. So they're called anti-parallel. Okay, now notice here that we have, again, two strands because here we have one nucleotide, phosphate sugar base on one side, and we have a long string of them bonded through covalent bonds. So covalent bonds are really strong bonds, and they link the phosphate and the sugar. These also link multiple nucleotides. So if this is one nucleotide here, it links it to this nucleotide down here, phosphate sugar base. This is one whole nucleotide, and it's linked to this nucleotide on top. These are linked by covalent bonds. These are really strong bonds. They're really hard to break. Okay. Now, notice here that our sugar has some pink numbers on it. what is this and why is it important? Well, we're actually going to need to number the carbons that make up our sugar, and we'll learn about why that's important later. But notice here that we have 1, 2, 3, 4, 5 carbons. Now, I know you can't see it, but there's actually a carbon here. So this is a molecule made up of carbon. If you were to look at it, you would see it if you were to look closely at the atoms that make up these molecules here. But carbon, 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 carbon. And we always number the carbon to the right of the oxygen as carbon number 1. And then we follow carbon 2, 3, 4, and 5. So this carbon kind of is like this chimney that's sticking up from the sugar here off of the carbon 4. Now, attached to this carbon 5 is our phosphate group. So the phosphate groups here are attached to our 5 carbon sugar. Sorry, 5 carbon is attached to the phosphate group up here. Now, our nitrogenous base is attached to our 1 carbon. So you can see here. Now, between the two nucleotides, we have our sugar connected to our next phosphate group on our 3rd carbon here. Okay, so we see that continues down. So here is our 5 carbon right here. And then here's our 3 carbon. So if you were to count, you would go 1, 2, 3. Okay, here's our phosphate group, 4, 5, and then our next phosphate group. Same thing down here. Now notice at the end of the molecule, if this 3 carbon is not attached to anything, it's just left with an OH group attached to that 3rd carbon. But if another phosphate were to come in here, it would attach to the 3rd carbon, this end here. Okay? Now, notice that this strand, these nucleotides here that are all connected on this left-hand side, the top ends with the 5 carbon sugar sticking up. And so we call this the 5' end. That's because the highest sugar on, or the highest carbon on this end is our 5 carbon sugar. So that's important. We call it the 5' end. Now at the bottom here, we call this our 3' end. And that's because the last carbon here is our 3rd carbon. So if something else were to be added on, it'd be added on to the 3rd carbon, right? It's the thing that's sticking out. Over here, the 5 carbon is sticking out. Now, if you look on the other side, this is antiparallel. It's going the opposite direction. So on top, we end with our 3rd carbon up here, but on the bottom here is our 5 carbon sticking out. Okay, so this, we say it goes from 5' end to 3'. That's how we read the molecule. There's like directionality to it, 5 to 3. This side, we start from 3' and we end with 5'. Okay, and you notice here that this molecule on the left-hand side, this string here of nucleotides is attached to the right-hand side. So we have 2 strands, 1 strand on the left, 1 strand on the right. So you have here our phosphate sugar base. Here's 1 nucleotide. And these bases are now going to be connected in the center. Now, notice that you have these dots here. These are representing something called hydrogen bonds. Now remember that hydrogen bonds are very weak bonds. They're not very weak, but they're weaker than covalent bonds, and they hold the bases together. You also have van der Waals interactions between stacked base pairs, so between this C and this A, that are holding the molecule together. So van der Waals forces are like kind of weak interactions between non-charged molecules that are interacting, kind of holding them together in place. Okay, now notice here that between T and A, we have 2 bonds, but between G and C, we have 3 bonds. So there's actually a stronger connection here between C's and G's compared to A's and T's, which only have 2 hydrogen bonds. Okay, now this structure is going to be so important for you to know. So make sure this is written down. Make sure you label the hydrogen bonds, the covalent bonds, the directionality with the numbers of the carbons, the phosphate group. We're going to be zooming in and out of this molecule, but you need to know that there are 2 strands. You need to know that on the outside, we have our sugar phosphate backbone. The inside are our bases that are paired up through hydrogen bonds. And you'll need to know that each one of these small pieces, these 3 here, are called 1 nucleotide. Now, the whole thing is called a nucleic acid, deoxyribonucleic acid, because the sugar here, its name is deoxyribose. So deoxyribonucleic acid. All right, now here's a simplified image. So back here we saw, here's our backbone, but if you wanted to not draw every atom, you could actually draw that sugar phosphate backbone as just this blue ribbon here. and that represents that sugar phosphate backbone. But we've included our bases, so A's and T's, C's and G's. So here's our nitrogenous bases. You can also just draw the sugar phosphate backbone. So a lot of times when you're just simply drawing the molecule, you'll just exclude the bases. You can also flatten the molecule, kind of like we saw before, and just look at the ladder. So if you took the sugar phosphate backbone and kind of stretched it out and flattened it into a ladder, you would see our phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Here's our backbone. Here's our 5' to 3' end. Okay, right? 5' because this is our 5-carbon sugar that's kind of sticking out, or sorry, 5-carbon that's sticking out on the sugar as like a little chimney here. And then down here are 1, 2, 3 carbon, right? And then here we have our 3 to 5. So we're running anti-parallel opposite directions. Here we are just showing the sugar phosphate backbone. We're not including all the molecules here as kind of ribbons. Here we are just looking at one side, okay, this dark side here, and we are using a lighter color to represent something called a more recently synthesized. So we'll learn more about this later, but DNA can actually copy itself. So if you were to separate these bases and to kind of pull that molecule apart and separate the strands, you can actually build a new strand on top of one of the old ones. And so if you build a new strand on top and make a copy of it, that's our more recently synthesized strand. So sometimes you'll draw like a lighter blue to show that that one just came in. And then sometimes you just draw the molecule as 2 straight lines, and you can label the directionality 5 to 3, 3 to 5. Now, last way you're going to see DNA represented is just a linear sequence of nucleotides. So when I say that, what I'm looking at is just the bases. Now I'm looking at 2 strands, right, because DNA is double-stranded. And here I'm looking at my 5 to 3' end and then 3 to 5' end. And again, we're just stripping everything down, so we're just looking at 5 to 3, so TGCA, and then the opposite ACGT. So again here, TGCA, ACGT. So it's the same exact thing, but we're just writing out those letters up for the bases and excluding the sugar phosphate backbone and all the detail. Okay, so we have already studied this, but here again are kind of similar pictures that we just looked at. Okay, so a couple more things. The directionality of DNA. You need to number the carbons, and I already taught you how to do this, but here is our sugar. Here's our ribose sugar. And if we were to, and again, remember, DNA is actually deoxyribose. So notice here that ribose looks a little bit different in that there's this extra oxygen. So all of the oxygens are attached to hydrogens, but with deoxyribose, you're going to actually lose one of these oxygens. So we'll come back to that. But let's say you just had a ribose molecule. This is a sugar molecule. You could show it, you could draw it out in one line here. So here's our carbons attached to oxygens, hydrogens. It's a sugar here. And then you could actually draw this oxygen. Okay, so you could actually detach this hydrogen. And if that hydrogen is detached, it can actually pair up. This oxygen can pair up with this carbon here, and it will form something called a ring. Okay, so there's a lot of detail on that. You don't need to know the details of this, just kind of show you that if these carbons kind of formed around each other in a circle, it would form this ring structure. Now, the carbon, again, that's right next to the oxygen, that's called our carbon number 1. And then 2, and then 3, and then 4. And then here's our little chimney. Here's our 5th carbon. So again, you don't see it because we get lazy and we don't want to draw all the carbons out. But here there would be a carbon, here carbon, carbon, carbon. And the lines in between are the bonds between each carbon. Okay, so that's how you read a sugar molecule. Okay, so again, this is really important. Here is our nitrogenous base. Here's our phosphate group. So first start with the oxygen. To the right of the oxygen, that's our carbon 1, and it's attached to our base. 2, 3, here's our 3', that's an important one. 4 and 5, here's our little chimney, and that chimney is attached to the phosphate group. Now this 3rd carbon, this oxygen, will again attach below to another phosphate group, but just keep in mind that this is the way you number the carbons. All right, so kind of again putting the whole thing together, we refer to the top part here as the 5' end because this 5 carbon is sticking out, the 3' end because this 3rd carbon is sticking out, and in between our 3rd carbon here, 1, 2, 3, is attached to this phosphate group, and it looks a little different here but here's our phosphate group kind of stretched out. And then 1, 2, 3, 4, 5. Here's our chimney 5 carbon attached to this phosphate group here. So in between two nucleotides, we have the third carbon and the fifth carbon are attached to this middle phosphate group in between. Okay, we're almost there. So we learned about these nitrogenous bases, and we know that they're paired up through hydrogen bonds. But if we look closely, we actually have names for these bases. Now, we have some categories that we're going to use. So the first is called purines. So purines would be adenine and guanine. So here's our adenine, or A, and here's the structure. Here's guanine and G. And just what you need to know about this is the purines have 2 rings. So A's and G's have 2 rings. I want to encourage you to think of a way to memorize this. Think of some mnemonic device, think of the word purine, and how you could remember that purines have 2 rings. I thought of a way when I was in high school to remember this, but I want to encourage you to think of your own. How can you remember that 2 rings are called purines? And these are attached to pyrimidines. So you will always see a purine attached to a pyrimidine. Say that in your head or say it out loud, pyrimidine, so you can memorize that word. Thymine and cytosine are called pyrimidines. And here's why. It's because they're made up of 1 ring. So 1 carbon skeleton ring. Now, A always pairs with T and C always pairs with G. Now, why is that? Well, here's a little helpful image. If you were to pair up 2 purines, so like let's say guanine and guanine, so the 2 rings, if you paired them up together, then the molecule would actually be too wide. Remember, Rosalind Franklin, her data showed what the structure of the measurements of the DNA molecule will look like. And so when Watson and Crick were putting together the molecule and trying to figure out how the bases fit together like a puzzle, if they put 2 purines together, the measurements didn't work. It was too wide. And why is that? Because those rings are too big. Pyrimidine and pyrimidine is too narrow for their ratios or their numbers. But if you took a purine and a pyrimidine, it's consistent with our X-ray crystallography or X-ray diffraction data that Rosalind Franklin took with her photo 51. So this shows us that purines and pyrimidines are always paired. Now, adenine will never pair with cytosine. So even though they seem like they could pair up, the chemistry doesn't work out. It needs to be A's and T's, G's and C's. And again, here you can see our hydrogen bonds. Remember, hydrogen bonds come from an electronegative atom like oxygen, kind of interacting with this hydrogen here. It's a weak bond. They're not fully covalently connected, but this is a weak bond here. And then we have our hydrogen bonds. So 2 bonds between A and T, 3 bonds between G and C. Okay, so make sure you know that. Okay, so we've already gone over this, but once again, we have our anti-parallel strands. They're parallel but opposite directions. And just to remind you that our phosphate to sugar, so here's our phosphate, here's our sugar, the bond. There's 2 bonds, and it involves carbons in the 3' and 5' position. So here's our 5' chimney kind of sticking out of this sugar here, and that's connected to this phosphate group. And then here's our 3' connected here, and it runs 5 to 3'. Okay, now on the end here, when you get to the very, very end, there's nothing attached, but here's our 3' So 1, 2, 3, and it's ending with this hydroxyl group or OH that has nothing else left on it. Here, our 3' end is on top and our 5' end is on the bottom. So this is the directionality. And the complementary means matching strands. They run in opposite directions. And then here's a little quote from Watson and Crick. They noticed that because the bases can pair up, it suggests something called a possible copying mechanism for the genetic material. So let's explain. Watson and Crick realized when they saw the structure of this molecule that because there's such consistency in how the bases match, maybe each of these could be copied. So like, let's say you took this apart, you could actually maybe create a new strand on top of one side by copying the DNA. We'll get into that. It's called DNA replication, but it kind of gave the scientists this hint that maybe because there's these connections here, that the DNA could be copied in some way and then passed down because of these rules, these base pairing rules. Okay, lastly, we already talked about this, but here's our hydrogen bonds. And these are weak bonds that can be broken easily to separate the 2 strands of DNA. And then, but between the sugar phosphate backbone, we have these really strong covalent bonds. They're called phosphodiester bonds, but really you need to know they're really strong covalent bonds. Okay, and then again, so the hydrogen bonds, you need to know that they are weaker. And this is helpful because if we were to cut these bonds, you would separate those two strands, and we're going to learn that you can actually copy the DNA if those strands are separated.